Hi, everyone, and welcome to this webinar about the Proximity Network Assay. And today in the panel, there is Peter Diali, who works as a business development manager for the European region for Pixelgen technologies. Myself, so my name is Michele Simonetti, and I work as a field application scientist at Pixelgen Technologies. I worked before also as a senior scientist in the R&D team, and I work in the development of the Proximity Network Assay. There is also Semjon Willier, who works as a clinician and scientist at the University of Fribourg. And he's going to present, after, let's say, an introduction about the assay, he's going to present some data that he generated using the Proxium kit. And we would like to thank him for inviting us. Also, Sentil, who works in his group, and worked in the generation of the data, as well as Marie Follo for organizing this webinar. So before we start digging into the method, it's always nice to give an introduction of why we wanted to develop this assay. And to do that, we have to look into the Semjon dogma of molecular biology, which of course everyone is familiar with, which start with DNA as the blueprint, RNA, manufacturing message, and the protein, important for, of course, cellular functions. So we know that most of the time when looking at protein, is what is observed is the presence of the protein and the abundance of the proteins. But we know that there is much more beyond that. We know that immune cells require efficient, precise, and reactive responses, and these responses are achieved through spatial reorganization of proteins. Therefore, it's important to introduce a new layer of omics, which is called protein interactomics, that basically studies how protein organization regulates cell functions, which we know is disrupted in diseases. And most of the time, when looking at proteins, there are multiple ways of doing that. If you're interested in looking at multiple proteins at the same time, so in the multiplexing, technologies, of course, like flow cytometry, mass cytometry, and also technologies with a more sequencing readout as a site-seq can, of course, help you with that. However, they are limited in the resolution. They're limited with the spatial information, let's say. On the other hand, if you are interested in the spatial resolution, technologies like microscopy are the gold standard. Super resolution microscopy is a perfect example, very high resolution, down to 20 nanometer, but limited in the number of markers that can be studied simultaneously. So there is this kind of gap here. This is what we wanted to fill in when we developed the Proximity Network Assay, which has very high multiplexing, 155 surface markers, very high resolution, down to 50 nanometer resolution, and you will be able to look at cells, at proteins on the cell surface in 3D. You will be getting more than 50,000 spatially localized protein molecules per cell in a high throughput manner. So all the markers can be studied in 1,000 cells per sample simultaneously. And together with the kit that I will show you how that works, we also offer a software which allows you to perform straightforward analysis. You will be looking at abundance of the proteins, but also spatial metrics like the clustering and colocalization. And the software is available for free. There is a lot of documentation online. There are also datasets that can be downloaded, and you can play with them, and you can have an idea of the kind of data output that you are able to generate. So how can we have this very high multiplexing and also very high resolution without having the drawback of microscopy? And the answer is that we don't use light to see where proteins are located, but what we have developed is a sequencing-based assay. And another important feature of the assay is the fact that there is no physical separation of the cells. All the cells are in bulk, floating in PCR tubes. Therefore, you are able to identify single cells, but also cell-to-cell complexes. So what we offer is a sample prep kit. There is no specialized equipment, which is required. It's very easy to run it in the lab, you only need a thermocycler to run the reactions and then a centrifuge to spin down and centrifuge the cells. And it's a library prep kit. You're able then to sequence your libraries with Illumina, for instance. It's compatible with all Illumina sequencers. The data processing and analysis part is done using the Pixelator software, which I described you earlier. And then I will show you how also the data output looks like. And this is then what you are able to generate. Since, as I said, there is no light to visualize where proteins are located, what basically happens is that we read DNA barcodes, and then the software plays this information on the surface of the cell. So here you see a single cell reconstructed, and what you see here, these are a B and a T cells that are interacting. So this is a summary of how the assay works, how the chemistry works. So as I said, all the cells are in bulk, floating in PCR tubes, and these are the main steps of the technology. So the first step is a PFA fixation step. This is done in order to immobilize the surface proteome. The second step is the addition of the barcoded antibodies. So as you can see here, each antibody has an oligo, and the oligo contain a barcode, which is a protein ID. So we have 155 different protein IDs, and also a unique molecular identifier, which makes unique each oligo and therefore each protein. You then add padlock probes, they bind into the oligo, they get circularized. So by doing that, you basically copy the information from the oligo, and then you're ready to perform a rolling circle amplification. So you elongate the oligos. You then add linkers. As you can see in the drawing, the linkers bind the rolling circle products that are in close proximity. And the key here is that the rolling circle product, it's basically an elongation of the oligo and contains multiple sites where the linker can bind. Therefore, it's not just a connection between one protein and one protein, but it's a connection between one protein and several different proteins, depending, of course, on the close proximity. So after NGS sequencing, this is, of course, a very simplified example of how the sequencing reads look like, but they contain the information of the two proteins that are in close proximity. And this is then what the software uses to reconstruct the location of the proteins on the cell surface, of course, in relation to each other. This was just an introduction of the chemistry, more a summary of the chemistry. Feel free to check our preprint, which is available online, which goes much more in depth into the chemistry and shows you also somehow, the kind of data output that can be generated using the Proximity Network Assay. If we look at the protein list, as I said, 155 proteins. So what we tried to do was including proteins that will give a high cell coverage. So for T cells, B cells, NK cells, monocytes, etc. Also wide selection of targets. So we included inhibitory receptors, adhesion proteins, activating receptors, metabolic regulators, and the panel is available online on our website, so feel free to check it out. And there are also controls. So we have included isotype controls, but also known protein complexes. If we look at the kind of data output that you're able to generate using the Proximity Network Assay, these are the three main different features. You are able to get abundance, of course. This is very straightforward. For each protein in each individual cells, you get an abundance score, which can be positive or negative. So low abundance or high abundance. But the beauty of the method is that in parallel with that, you also get spatial metrics, like the clustering and the colocalization. So abundance is very straightforward. This is an example of a UMAP, which is generated from a PBMC sample. So you see all the different subpopulations here. The beauty of this is that you can go into each of these dots, which are basically the cells, and you can look at the abundance, but also the distribution of all the 155 markers. This is just an example of CD45 in a naive CD4 T cell. So this is what you're able to generate What you see here, these are the subpopulations. These are all the 155 different markers. And in red, basically, these are the different levels of expression of that marker, so the abundance level. There is also another way of plotting the data. This is very familiar to, of course, to the people that are using flow cytometry. But what you see here, we basically plotted CD4 and CD25 to identify activated T cells. And this is something that's very similar to what you will get with flow cytometry. Of course, the big advantage of our technology is that you can do this kind of plot with any of the 155 targets that are included in the kit. If we move to the first of the spatial metrics, so the protein clustering, what you see here is basically what you're able to observe. This is what the software generates, which is a score, which in this case is called clustering score. So what happens is that the software finds all the different networks of informations. These are the single cells. And of course, each network contains nodes and edges. So what happens is that the software reshuffles all these nodes and edges, creating a normal distribution. And then it looks for how many times one protein, of course, is closer to itself more often than it is by random chance. So basically, it's correcting everything based on the abundance of that protein. So that gives you a score, which is positive if, as you can see in the drawing, that protein is clustering on the cell surface, or close to 0 if that protein is dispersed on the cell surface. To show you how that works, we can look at the antibody-drug mode of action of rituximab, which induces the clustering of CD20 on B-cell cancers, enabling the binding of NK cells. So what happens is that what you see here, this is the protein clustering. If we look at CD20 on the treated cells, you can see that we detect higher score compared to the control or compared to another marker which is not affected by that. And what you see here, this is the confirmation. And we used here fluorescence microscopy. So to the left, you see the control with CD20 dispersed on the cell surface. And in this case, on the stimulated cells, CD20 clustered on the cell surface. Now imagine by doing that, but in a pairwise manner. So now you look at pairs of proteins. So this is the protein colocalization, and you will be getting a colocalization score. So that score is positive when the two proteins are colocalizing, close to 0 if they are randomly dispersed on the cell surface, but this score can also be negative if the two proteins are segregating from each other. So you can imagine that this can happen, for instance, if you study immune cell migration. In that case, an immune cell forms a front and a back end, and you can see segregation between proteins. Or for instance, also to identify cells that are interacting. You can imagine that if you have a B and a T cells that are interacting, they have specific markers. And this is how actually you can use the score to identify which are the two cells that are interacting. Now, this is very straightforward, of course. Since it's a pairwise measurement, you start identifying protein complexes. But since, as I said before, you're not just comparing one protein to one protein, you are comparing 155 proteins to 155 proteins. You're able to generate colocalization heat maps. So what you see here, this is a selection of 39 markers. And in this case, we are looking at activated T cells. Most of the times, you don't need to plot all the 155 markers. You're just plotting the markers, of course, that are more informative, or the markers where you get, of course, hints of colocalization. But what you see here in the different colors, in red means that proteins are colocalizing. In blue means that it's segregating from each other. So all these squares here, these are multi-protein domains. You can, of course, zoom in, and you can look at the different markers that are part of the domain. In this case, these are well-known protein complexes. But to show you how that works, we can look at these three proteins here. This is CD55, CD48, and CD7. So our software detected colocalization between CD55 and 48. So what you can see in the super resolution microscopy images is that indeed CD55 and 48 are present at the exact location in this kind of foci on the cell surface, whereas CD7 is excluded from that portion of the cell surface, is more randomly dispersed.
So
why using Pixelgen for your research? So I'll show you now some examples, but in general, the technology can be used for in-depth disease profiling, to reveal new molecular mechanisms, for drug target identification mode of action. We have seen an example with rituximab before, but also for biomarker discovery and patient stratification. So the Plex is designed for immune cells. And of course, immune cells are involved probably in all the diseases. Therefore, as you can imagine, it's very broad, the applications of this assay. So can be used in hematology, immuno-oncology, immunotherapy, autoimmunity, and basic immunology studies. To show you some examples, we can start with immunotherapy. And of course, one field of research where our assay can be super useful is in CAR-T research. Because we know that when, for instance, creating a new CAR-T cell, non-optimal organization of the receptor will have a large effect on the cell function and also exhaustion of the cells. So understanding and studying actually the architecture and the interactions, the colocalization between the proteins of the CAR-T cells can enable the development of more effective therapeutic products. So the CAR-T cells can be easily identified among PBMC cells. You can see in the zoom up here, you see the CAR-T cells here. Something that can be added, we can also talk about this later, but something that can be added in our Plex is two barcoded antibodies that will allow you to tag the CAR-T cells. And one is the one that we have used here, that you see here. This is an FMC63 barcoded antibodies. So it's to detect anti-CD19 CARs. But we also offer a flag tag. This is more universal as a construct. But in this case, as you can see, the FMC63, which is dispersed on the cell surface, as expected. But what can also be done is really to look at the interactome. So all the different proteins that are co-localizing. So with these big co-localization heat maps. But if you are interested in the CAR receptor, you can, of course, zoom in. You can look at the different interaction partners. In this case, you can see a lot of addition molecules, like the ICAMs, for example. But you can also see what's segregating from the marker. In this case, it's lipid draft. And this is something that's common with TCR, alpha beta, which, by the way, was also co-localizing with the receptor, telling you probably that they share some spatial patterns together. But you can also build functional assays. So what you see here, these are CD19 targeting CAR-T cells that are co-cultured with CD19 Raji cells. You can see that you have the T cell alone and two different time points, 4 hours and 24 hours of co-cultures. And then the cells were basically, the samples were run using our assay. So what you see here, these are changes in abundance, and we are comparing the time points to the T cell alone. So red means that you have a higher abundance of the specific marker in the time point compared to the T cell alone. So you can see that after 4 hours, you start having some markers that start popping up. After 24 hours, a lot of markers that are important for sustained activity of the cells, but also exhaustion are present. And you can also look and point towards the fact that, for instance, the CAR receptor has less abundance compared to in the time points. And this might tell you that, for instance, there is either internalization of the marker or there is simply masking due to cell-to-cell interaction. But again, this is only looking at abundance. Of course, with our technology, we can go beyond that. So something that we have observed is actually also exchange of information between cells. So what we had here was, this is a UMAP. You can see all the different samples that we had, so the CAR-T only, and also the two different co-culture time points, the 4 hours and the 24 hours. And we looked at the abundance of ICAM1, which we observed was quite abundant in the co-cultured cells. So we started looking at the spatial organization and where the marker was present on the cell surface.
And we noticed that ICAM1 on the T cells actually was not dispersed on the cell surface, but was actually clustering, was present only on a certain portion of the cell membrane. And actually, the same exact region, there were also B cell markers. And again, we were looking at T cells. And T cell specific markers were excluded from that region of the cell surface. So actually, we were not looking at T cells that were overexpressing ICAM, but we were looking at T cells that actually gained pieces from the B cells during interaction. So this is a process which is called trogocytosis. And for instance, can happen when there is killing of the cells simply. And the software is able to identify, we call them tumor patches. So in this case, you have 4 different cells with either no tumor patches, 1, 2, 3 patches, and you can quantify this. So what you see here, you have the T cell alone as a control, and then you have the 24-hour co-culture, the different colors, the dark gray, these are the cells with unactivated profiles. Of course, they have more tumor patches, but very interesting, cells with a more exhaustion profile, they were the ones with more tumor patches, indicating a correlation probably between the exhaustion profile of the cells with the, we can say, the cell killing, probably the trogocytosis more in general. So this is just an example of how the technology can be used in CAR-T. Another application can be in hematology. So here I want to show you one application in AML, MDS. Here we used it to look at responders versus no responders. And actually, this was in bone marrow samples. And the idea was to identify also pre-treatment response biomarkers. This was in collaboration with Aaron Vini lab at Columbia University. So the background is that a large fraction of AML patients do not respond to treatment with hypomethylating agents, which are very effective, but only in 20 to 20% of the cases. So it will be very important to predict which patients will respond. So in this first pilot study, we had 5 responders and 6 no-responders. So we used all the features that you basically get using our technology, so abundance, but also the spatial metrics. So we trained a random forest classifier, and then we used that to predict on the remaining cells if they belong to either the responder or the no-responder groups. So when looking at abundance, the first thing that can be noticed is that the no-responder group have, on average, more AML blasts prior to treatment, but this is not enough to predict individual responses. So what we did was really looking also at the spatial metrics, using them to train our software, let's say, the random forest classifier, and this actually gave an accuracy of 94%. And actually what we identified was that there were non-responder specific features of protein organization that might present new type of therapeutic targets. So this is just one way of how actually the technology can be used also for patient stratification, for instance, or to understand the differences between responder and no-responders. But something that can also be done is to study drug resistance and relapse. This is something that we did in CLL. And also to understand resistance mechanisms. This was in collaboration with Richard Rosenquist's Brandel lab at Karolinska Institutet. And this study was divided into different parts. In the first part, we basically wanted to understand which were the mechanisms behind the drug resistance in CLL. So what we had was CLL cells, which were treated with 3 different drugs, either BTK inhibitor or BCL2 inhibitor, to induce resistance. And then we looked at the differences on the proteome organization. So what you see here, this is basically a colocalization heat map. This is what I described you before. So the markers here are the markers with increased colocalization. The markers here are the markers with decreased colocalization. So immediately you can see that there are certain features that are in common, but also that there are differences between the different drugs. And something that we observe to be in common is actually the fact that the resistance to BTK and BCL2 inhibitors was associated with altered CD19 thread-spanning colocalization. So it can be used actually to uncover drug-specific resistance mechanisms. To go a bit beyond that, we also used our assay to actually monitor blood composition, which of course it's key to understand the disease development of CLL. So this is something that can be done using our technologies. This is just an example, something that you would get very similar to flow cytometry. You can get in this case for CD8 and CD3, but again, you can do this for all the 155 targets there. But you can use the assay actually with great accuracy to monitor the blood composition. So in another part of the study, actually, we wanted to see if the method can be used to identify novel drug targets in relapsing CLL. And actually, what are the differences in the protein clustering compared to in the relapsing CLL cells compared to the CLL in remission? So the background is that CLL actually is marked by expansion of cancerous lymphocytes, and identifying proteins and pathways that are involved in the disease relapse can aid to the discovery of new druggable targets. So the question is can protein organization reveal new biomarkers of relapsing CLL cell disease? So what you see here, you have the percentage of CLL cells. You see that the samples were collected in 3 different time points. You see before treatment, you see in remission, and then you see in relapse. So what you see here is the comparison between abundance and clustering. And what you see here, you see relapsing CLL cells compared to CLL in remission. And in this square here, these are markers that have higher abundance and clustering in relapsing CLL cells. So we can, for instance, look at CD352, which is LAMF6, which is a regulator of B cell survival that was detected to spatially cluster in relapsing CLL cells. And as you can see in the spinning cell here, in yellow, you can see the clustering of the marker. So you can speculate, for example, that abrogating SLAMF6 reorganization might represent a potential therapeutic intervention in relapsing CLL cells. So I want to show you one final example in hematology, how actually the method can be used. And one application that we had was to profile immune cells. In this case was lymphoma profiling, comparing barked lymphoma cell lines to healthy B cells. This was in collaboration with Illumina. And the background here is that we wanted to explore the mechanisms that are behind the drug response in barked lymphoma. And one important marker is CD40. And CD40 targeting antibodies induce proliferation in healthy B cells. But on the contrary, the same treatment actually induces apoptosis in cancer cells. So it would be very important to understand if there are spatial differences that might explain, for instance, this different response. So something that we did was profiling healthy cells, but also barked lymphoma cell lines. As you can see here, these are colocalization heat maps. You can zoom in this multi-protein domain, which is lost in the barked lymphoma cell lines. And actually, you can notice that CD40 actually was present in this domain. So CD40 was actually part of, this is a tetraspanin-rich domain. So in healthy B cells, CD40 was part of that domain, but actually was excluded in all the barked lymphoma cell lines. So actually, you can speculate that the segregation of CD40 for that domain actually potentially is regulating the signaling behavior. And something that we did in the same data set was also identifying bispecific drug targets using actually protein interactomics. So related to this, it's important to know, of course, that potential tumor targets are often also expressed on healthy cells. So identifying cancer-expressed protein pairs, which are of course specific, might represent potential targets for base-specific therapies. So something that we identified was that CD38 and CD37, they had increased colocalization, for example, in the 3 different cancer cell lines, but not in the healthy B cells. So of course, this cancer-specific localization has the potential to enhance bispecific drug specificity and to minimize off-target effects. So to conclude, here is a long list of applications. I just show you 3 different examples, but of course we have seen some examples in hematology, immunotherapy and cell engineering with CAR T cells, but this can also be used in basic immunology, autoimmunity and inflammation, also drug development and pharma. So to recap, the kit actually, as we have seen when Pete actually introduced it to you, this is a sample prep kit. So this is what we basically offer. So the kit is for 8 reactions, so 8 samples, up to 1,000 cells per reaction. And together with the kit, we also offer for free a software, which is called Pixelgen. And there is a lot of documentation online. There is also a dataset that can be downloaded. You can play with those. You can have an idea of the kind of output that you're able to generate. And I really want to stress again the fact that there is no specialized instrumentation which is needed. So the only thing that you need in the lab to run it is just a thermocycler to run that basically holds PCR tube and a centrifuge to spin down the cells. That's basically all that you need to run it in the lab. But with this, thank you for attention. And I think, give the floor again to Pete, probably, to introduce Semjon.
Yes, thank you so much, Michele. And good to see you've got a good internet connection, Semjon, because I've been used to losing you in recent days. So good to see you're here with us. So I think finishing up on that B cell data was quite nice, because now we're going to transfer to some, as I mentioned, hot off the press data from Semjon, and that has been analysed by our own in-house scientists, and as you see here, Senthil Ramamanathy. So I'll leave it with you to carry on. Thank you very much, Semjon.
Well, thank you very much, Pete. So it's a pleasure to give you a short perspective from our experience with Pixelgen, and perhaps as a kind of introducer, I met Pete at the meeting in Munich this year in February, and I was very intrigued by this nice data that Michele just showed on this B cell malignancies, and I thought, well, what could be done for B cells? Why couldn't it be done for T cells? And so this is why I'm going to talk to you for just a few minutes and give you some snapshots about what we have been doing, in particular looking at T cell malignancies and how we can kind of get our fingers into the differences between physiologic and malignant T cells. And as Pete has just said, the data has been produced. Actually, I missed to mention Eileen Balion, who has been the technical lead in this endeavor, and analyzed by Senthil, but also in close collaboration with the team from Pixelgen, including Stefan, who has helped here quite a lot. So just very short, because I think it's mainly the purpose is to give you an idea about this technology. We have recently identified CD28 as being highly overexpressed. If we look at malignant T cells in comparison to healthy T cell progenitors, this is a little expression, some expression data from patient materials. I don't want to go into much detail, so this is the reason why we're interested in that. And if you look here, we could also show just recently that if we are looking that we are stimulating some malignant T cells with CD28 antibodies, so agonistic antibodies, these cells can grow better under certain conditions detailed here. So we think that CD28 might be an interesting target for patients with T-ALL, and it might even bear some functional relevance. So this is why we're interested in that. And as I just said, really the aim of this little project was to detect surface features that are different between malignant and physiological T cells. And the main focus was here on CD7 and CD28 as some targets that we're currently investigating in the lab. So this is the reason why we took some malignant Jurkat cells and also some physiologic cells in order to understand in particular these details, looking at the different layers of information that Michele has just beautifully laid out for you, which one of them would be abundance. And this is just a short snapshot that should show you that what you can do with flow cytometry, you can do easily also with the Pixelgen kit. So you can look at just differences in abundance if you look at T cell activation. And I'm very sure that many people in the audience will be aware of several of these antigens, and some of these are canonical molecules that are upregulated upon CD28, CD3 co-stimulation, and some other markers are quite to be expected to be downregulated if these cells proliferate for a few days. So this kind of helped me as a flow cytometry person to believe the data that we have generated before. And now perhaps to look a little bit more into detail, if we look at differential abundance in the CD28 knockout Jurkat cells, comparing them to CD28 wild-type Jurkat cells, I think it was, to me, very interesting. I mean, first, obviously, to see CD28 being the molecule that has been downregulated the most, but then again, to see some features, some of these 155 surface markers to be upregulated. And if you think a little bit more in detail about it, you can make some reason out of it. In particular, if you think about some adhesion molecules that are LFA1, for example, or CD2 that are upregulated in response to CD28 knockout, this seems very interesting if you think about CD28 also as a molecule that structures cell surfaces, also co-stimulation molecules, which is also CD2, but also DNAM1 or CD226, which could kind of recompensate. So this is our theory, could recompensate the lack of a stimulatory molecule or co-stimulatory molecule on the cell surface, and also to observe HLA-DR as an alternate activation marker, which might be an indicator or an epiphenomenon of alternative activation processes within the cells that have lost CD28 as a co-stimulatory marker. Then this was just the abundance, and now we're looking at the differential proximity or the proxiome of these cells. You will appreciate in this plot that only details those top 15 markers that are differentially showing differential proximity out of the 155, and you can easily see that most of the markers that show changes here in the proximity or most of the proximity that are changed are proximities that incorporate CD28. And however, there are two interactions that are altered, which is the CD2 and CD82 reduced proximity, and the increased HLA-ABC CD43 proximity, which might be interesting and shed some light onto what happens on the cell surface once CD28 has been lost. In particular, CD82 that helps to organize the plasma membrane and can cluster with co-stimulatory and adhesion molecules such as CD2. This, I think, is a very interesting feature to look at, because if you then go on and if you feed this data into a string analysis, it is not super surprising that you could think about, and perhaps I make this a little bit bigger, you could think about CD28 as you can see here as a linker that enables interaction of CD82 to CD2 in a preliminary data set, nothing that we would now start to postulate, but still something that is interesting and that gives us some better understanding of what's happening if these target antigens are present or not present on the cell surfaces. Also, you can feed the output of the differential proximity into gene cell enrichment analysis, and you will see which downstream signaling pathways will be changed upon CD28 knockout. What I also found very interesting is the visualization of the proximity in absolute ways, because just now what I've shown you is the differential proximity, and here this is the absolute proxiome, you could say. So I learned a lot of new words when I worked with Pixelgen data, and I like proxiome very much. And you can see here that there are some Jurkat cells, and you see which other cell surface markers are co-localized, how strongly. If you look at the breadth or the width and the color, you can see the direction whether it's co-localized red or it's not co-localized, it's mutually exclusive, then it's blue. And you can see that there are some very specific differences in this malignant T cell when you compare it to a resting and to an activated T cell. And I think some of these differences might be very interesting if you look, for example, co-expression with CD50 or co-expression with CD352. These are features that are really differentiating for these limited samples, at least physiologic from malignant T cells. So I think this is very interesting, in particular what just Michele has alluded to, thinking about bispecific approaches to kind of differentiate what is malignant and what we want to get rid of and attack with them with therapy such as bispecific agents or what is part of physiology. And then last but not least, we're also working on CD7 as the primary TLL immunotherapy target at the moment. And here, if we look only at the proximity score of the proxiome of CD7, again, this is absolute data. You can see again here the Jurkat cells, so malignant T cells. You can see I'm resting T cells and activated T cells. This is the same visualization, but you don't see a lot of co-expression patterns, in particular if you look at the CD7 proxiome in the Jurkat cells. But then if you look at and what you also see, obviously, is that upon activation of T cells, a lot of changes can be observed in the proxiome of CD7. But this is not my main point. If you look at a different way of this proxiome score, of this proxiome data for CD7, you can also appreciate here that if you see that you will see that CD7 is, for example, here is clustered a lot in the activated T cells, but it is not clustered in the Jurkat cells. So, and you can also appreciate that there, if you look at this Venn diagram here, that there are several features that are really differentiating that are differentiating healthy, activated, and resting T cells from malignant T cells, which I'm not going to mention here, but which just kind of illustrates the multitude of analyses and the detail that you can look at these cells with the Pixelgen data. And so, yeah, I think I'm not going to read off these markers to you, but the main point is really that you see some of the Jurkat cells do not to show some specific differences when it comes both to activated and resting T cells that are, for example, shown here, the segregation for CD45RA and CD19, which is not observed in any of the other physiologic T cell conditions. And now, if you think about what Michele has shown us in the beginning, I think it's very interesting to see that you see this the CD7 clustering to a much lesser degree, as I've just shown you. Also in this 3D rendering, which is one individual Jurkat cell, and we can look, as Michele said already, at every individual cell when it comes to the CD7 clustering on the cell surface. And if you compare this to activated T cells, where we just have seen that the clustering is much higher, you can appreciate that the lighter spots here indicating probes that have or reads that have identified CD7 to be expressed are more localized here in this sample. So for me, this has taken a few months, but I think it's a very interesting technology that comes at the price of not having the data right away if you finish your flow cytometry analysis, but you have to you have to go through the NGS analysis. But then I think it's really some features that you just cannot get your hands on with any other technology, I think, at the moment. So, and also, I mean, I can just confirm what Michele and Pete have alluded to, that the necessities, I mean, you need the kit. This is for sure. But other than that, it's just basic lab resources and a sequencing provider. And then you can generate this kind of data. So that was it from my part. And I think I will go back to Pete with that.
Thank you so much, Semjon. That's really cool. Really nice to see that data and very clearly presented. So thank you so much. With that, we can open up to questions. I'm not seeing that anyone's typed anything in the chat, so either there's something stopping people type or somebody's not got questions. So if somebody does have a question and they try typing, I'm also going to attempt to allow everyone to talk, which I have to do one at a time by the looks of this. But if anyone needs to speak, I'm just clicking through all the names now. Still nothing.
Peter, can you hear me?
Orion. Oh, yes.
Now we
Hey.
hear somebody.
Beautiful data. I've actually a question to Semjon's data. I think the data looks really beautiful, and I think it nicely illustrates, the commonalities and dissimilarities between like the Jurkat and different T cells used. Do you also see some heterogeneity within like one of the sample groups between like with regards to the molecule co-localization and polarization? Or like are most of them like, for instance, for the primary T cell, whether you see like differences in in between the co-localization of different cells?
I think, I mean, I absolutely get your point. To be very honest, we just had within the last 2 weeks, we had our relevant meetings to kind of wrap things up. So I think this is an absolutely, this is a very valid point that we currently just looking at p-values and looking at medians and median differences. So this is obviously something that we haven't digged so much into at the moment, but which we absolutely have to in order to kind of, before we go forward, to really think about new hypotheses for new therapies and so for me, I can only say we are not there yet, but this will for sure be something that we have to address before we kind of wrap things up and go forward with the next experiments. Thanks.
Because I think that's really like the advantage of the technology, right? That you have individual cells, and if you have like a, one gradient of polarization, you will be able to capture this whole gradient, and I think it may help you to understand like how, for instance, now the CD28 signaling may exert its role on membrane protein complexes.
So I would guess that kind of the things that pop up, because this is all so actually this is something that Sentinel has been doing from our part, but I guess that the like those differences that I've just shown you, if they kind of are above the threshold of significance, as far as I've understood the data or the documentation on the pixelator, this would indicate that probably there's not such a high variety that this kind of would compromise the statistical significance. But still, if you think about going forward with a difference that you have observed in the proxyome, you better be sure that this is something that has a clear cut between health and malady in order to not develop something that doesn't help so much more.
Yeah, perfect. Thanks.
So if anyone else wants to speak, I think they need to unmute themselves, but I believe everyone else is able to speak now if there are other questions. And I don't think I saw anything in the chat box still, but let me double check. No. So in that case, just thank you to Michele and Semjon. I know I said I was going to record it at the beginning, but I noticed partway through it had stopped recording. So we might repeat internally the first few slides and join it together for you. Obviously, if you're not PIs yourselves and your PIs are interested in learning more, we're more than happy to present again to them. And if you are PIs yourself and have questions, or whatever stage you are in your career, please reach out. It's our job to talk to you and share what we know with you. So never be shy to get in touch. Thanks so much, everyone, for your time and attending.
Thank you. Bye-bye.
Bye-bye, everyone. Cheers.